Alrighty. Hope everybody had a good weekend. Today, oh, got 0 viewers. Perfect. Today we are going to be looking at evolutionary scale prediction of atomic protein structure with a language model. So this is a paper coming out of Facebook research. So this is kind of Meta's answer to the DeepMind protein folding, trying to position itself as also a contender in that race of using deep learning for biological use cases. This is the GitHub repo for it. So we can actually look here. Let's look at the insights. Contributors. Okay, so we have Tom Sersu, Joe Shim. Okay. Tom Sersu has to be on here. Yep. There he is. So that's actually interesting. Usually the kind of contributor is usually around the top here. It might be that these people here are the people who maybe wrote the paper, made the stuff. A lot of kind of disparate people making little commits here and there. Really, most of this work comes from this guy. Follow him. Perfect. So going back to the paper, I have this open in PaperPile, which is my kind of favorite paper management tool. It allows you to kind of easily add papers and works across your devices. Alright, so let's get into it. Artificial has the potential proteins to extend protein structure to 200 million catalog proteins. Okay, so that's kind of, I guess, roughly the size of the data set. Exponentially growing billions of protein sequences revealed by large-scale gene sequencing experiments. Direct inference of structure from primary sequence using a large language model. Okay. Drinking some espresso coffee right here. Okay, train models up to 15 billion parameters. Largest language models of proteins to date. Okay. This is a three-dimensional structure of a protein. They say faster. Okay. Look at little Boo. She's interested in the cursor. Can you see her? Boo, you need to let me review this paper, okay? $225 million high conference predictions, including millions whose structures are novel in comparison with experimentally determined structures. $617 million structures. 200 million, 225 million, okay. Boo, that's just the camera, okay? You need to find something else to do. There you go. Okay, so you have a couple different teams here. You have New York University, Stanford University, MIT. Okay, that's pretty big collaboration there. Sequences of proteins at the scale of evolution, biological structure. Okay. Structure and function can be inferred from the patterns in sequences. Yeah. Okay. I like these citations. I think these are bio citations, not square brackets. It's parentheses and then you dash. So you wouldn't say 1, 2, and 3. You just say 1 through 3. Okay, we have, of course, Shannon's model for entropy. This is kind of a linchpin of information theory and a lot of statistics. And behind a lot of these language models is some kind of mathematical construct that traces its roots to this kind of information theory and entropy. Increasing. Yep. Large-scale attention-based architectures. That's the transformer architectures have kind of taken over in LLMs. They're pretty much everywhere. You take these same LLMs and you train them on amino acid sequences. Yeah, I think the problem here is that, right, amino acids are three-dimensional. Anything in biology, right, is three-dimensional, but large language models, they're trained on two-dimensional sequences, right? Or even one-dimensional sequence where a sentence is kind of has a starting point and then an end point, and there's time that progresses through it. But there isn't that same thing inside an amino acid or a protein, right? It's like a 3D structure. So the way that they feed these 3D structures into these LLMs, I think, is still some kind of hard-coded crap that someone came up with, where they take like the letters and then they have this like kind of like tokenization of the protein. But I wonder if anybody's going to actually come up with something better than that. Yep, emergent capabilities. Greater capabilities emerging as computation data and the number of parameters, right? So the bigger your model, the longer you train it for, on the bigger the data set, the better it's going to be. Yeah, this is the coolest part. Raise the possibility that a parallel form of emergence might be exhibited by language models trained on protein sequences. So it's impressive when LLMs have this kind of uh, emergent intelligence on text, and humans are very good at text, right? We think in words, we think in sentences, so we can look at the emergent intelligence that LLMs have on sentences and understand it and kind of think about it and judge it and quantify it. But humans are a lot less good at understanding protein sequences or even even knowing what that even means, right? So it's interesting to think about what when these LLMs inevitably have emergent capabilities on protein sequences, like what does that even mean? Do they understand patterns that we don't understand, and how do we know that they've understood something that we ourselves do not know? Yeah, so the task of filling in amino acids and protein sequences. So this might be the kind of big contribution in this paper. I think that the way that DeepMind approached this problem is it's very much kind of like a one-to-one supervised learning task where they have kind of the components of a protein or a molecule, and then they have the 3D structure and they kind of predict it. But Meta here is doing a filling in the missing assets. So it's almost like uh, I would say it riffs more with the kind of next token prediction that is used to train these LLMs to begin with, where they kind of give it a sentence and they say what's the next word in that sentence. Boo, you looking at this cursor? Look at that cursor. Yeah, so they have a couple different models, right? But these kind of in the modern world of deep learning where you've got these massive parameter or these massive models, kind of it's become standard in these type of research efforts to have a smaller models and bigger models, right? And I think that's what they're saying here is that their small model is 8 million parameters and their big model is 15 billion. The language model's understanding of the protein sequence perplexity. So perplexity is kind of what they're going to use to quantify how well the model understands the protein sequence. End-to-end atomic resolution structure prediction directly from sequence. Boo likes to bury my coffee because she thinks it probably smells like poo because it's just like burnt bean taste. Okay, so eliminates the need for a multiple sequence alignment. Okay, so it sounds like they might be doing something interesting with the inference. Maybe similar to kind of how a lot of bounding box detection and computer vision stuff originally, these networks had kind of multiple passes, right? You have the region proposal network, you have the kind of different classification heads, and like there's this kind of multi-step process to going from an image to a bounding box. So there might have been something similar in this type of problem where there's kind of multiple networks and multiple steps, and they might be doing something to simplify that process, right? Greatly simplifying the neural network used for inference. Okay. Can take over 10 minutes. Okay, so apparently AlphaFold can take up to 10 minutes to predict. What is metagenomic proteins? Metagenomics sequences knowledge of protein, metagenomic datasets. Okay, so I think this is basically when they sequence a bunch of DNA where they don't really necessarily know, right? They take like some pond water and then they get all the DNA out of that, and that's going to be DNA from bacteria, DNA from tadpoles, DNA from plants, and they just kind of sequence all of it. And they say we don't really know where this this genome comes from, but we're just going to sequence it and these little snippets. And I think this is kind of yeah, these efforts have contributed to an exponential growth in the size of protein sequence database. Yeah. Billions of proteins. Okay, so human have about 20K proteins, and then you have about 200 million in UniProt. Folding practically all sequences is in mgniffy 90, over 617 proteins. What is mGiffy 90? Mgiffiny 90? Did I even spell that right? mgniffy 90. mgniffy 90. Magnify 90. mgniffy 90 dataset. Magnify, it's a UK database here. Magnify, the microbiome analysis resource. About us. European Bioinformatics Institute. Okay, so it's kind of like some open Alzheimer's research, cancer research. Okay, so it's like an open database of genomes done, kind of funded through European governments and cancer and kind of Alzheimer's research. Okay. Able to complete this characterization in 2 weeks on a heterogeneous cluster of 2,000 GPUs. Right, so they had a couple different GPU types. High confidence predictions made for over 225 million structures, revealing characterizing regions of metagenomic space distant from existing knowledge, with the vast majority of high confidence predictions being separate from UniRef 90. Okay, what is UniRef 90 dataset? Sorry about that. UniProt reference clusters provides clustered sets of sequences from the UniProt knowledge base. Okay. So this is like about comprehensive resource for protein sequence and annotation data. UniProt funding. Okay, so funding is National Human Genome Research Institute. A bunch of government money. NIH, National Institute of Health. This is the UK socialized medicine. Okay. Operations can be accessed. Okay, I need to go take a break here, so we will return shortly. Be right back. Put this on here. Be right back. I'll be back. we're back. Sorry about that. Alrighty, close out of that. Go back to the paper. All predictions can be accessed in the ESM Metastage Economic Atlas. Explore. Holy crap, this is all the proteins in the world. Protein Data Bank. This is a 1 OMS E. coli trigger factor binding at the ribosomal exit tunnel. The exit tunnel region of the ribosome is well established as a focal point for interaction between the components that guide the fate of nascent polypeptides. So this is the actual sequence. Hydropathy. So maybe how hydrophobic different parts of the sequence are. Right, so if you have some parts of it that are more hydrophilic, some that are more hydrophobic, that's going to kind of influence the way that it folds. It's kind of cool. What is this purple versus, oh, it's down here, unknown versus known. So interestingly, right, you have regions here where you have a ton of unknown, and then you have some regions here where you have a ton of known. So what is this known region here, for example? View structure. Damn, and look at that, this little 3D visualization, and you can look at the individual sequences here. So this little part, what is this little edge here? Right, so P, N, P, L, L, N, this is fucking badass. This is so much information here. And these are actual atoms here. That's pretty cool. Let's put that in the description here. I'm going to put that there as well. We're also going to call this ESM. Alright, let's get back to the paper. We begin a study of the emergence of high-resolution protein structure. A new family of transformer protein language models, ESM2. Okay, 8 million as for the small one, 15 billion for the large one. Previous generation model, ESM1. ESM2 improvements in architecture, training parameters, and computational resources. So bigger, better, badder. Okay. Okay, trained with the masked language modeling objective, right? Yeah, so you basically mask out part of the protein structure and then try to train the model to predict the missing part. Yep, simple and unsupervised. I think this is important because you can just take an existing database of proteins and then just kind of infinitely create training data from that. Unsupervised learning is really the meat of the cake. if you want to use the cake analogy. During training, sequences are sampled with even weighting across 43 million UniRef 50 training clusters, 138 million UniRef 90 sequences. So total size of the data set is 650 million or 65 million unique sequences. Large improvements in the fidelity of its modeling of protein structures. This fidelity can be measured using perplexity, which ranges from 1 for a perfect model to 20 for a model that makes predictions at random. Perplexity describes the number of amino acids the model is choosing between for each prediction. Okay, so 8 million parameter model has a perplexity of 10, 15 reaches a perplexity of 6.37. Is there in information theory, perplexity is a measurement of how well a probability distribution or probability model predicts a sample. A low perplexity indicates that the probability distribution is good at predicting the sample. The perplexity pp of a discrete probability distribution p is defined as the multiply over all x, the probability of x times the negative probability of x. H of p is the entropy of the distribution, and x ranges over events. Transformer models trained with masked language modeling are known to develop attention patterns that correspond to the residue-residue contact map of the proteins. Sequences with high evolutionary depth saturate at lower model scales. What is evolutionary depth? I don't know. Maybe they made that up. Maybe it's just how many different proteins are related, like how many are in the cluster. How similar are the different things that are similar? At each step, there's an overall shift in the distribution. Yeah, so this is kind of interesting here, right? Nonlinear improvements in the accuracy, and you notice this in LLMs too. Sometimes they call this inflection points, emergence points, or something like that. But basically, this happens in reinforcement learning too, where you'll have kind of a reward and it kind of peaks, and then suddenly there's like it kind of stays the same for a bunch of steps, and then randomly it'll jump up and go to something else. And it's kind of the analogy, the anthropomorphization analogy here is that the model is like once it gets it, once it finds right kind of the edge of a kind of a huge ditch in loss space, it'll quickly fall into it and get to a new local optimum. Figure 1D plots the change in the distribution of long-range contact position at each transition to a higher level of scale. The accuracy of contact map prediction and perplexity are linked, with proteins undergoing large changes in contact map accuracy also undergoing large changes in perplexity. We project out spatial coordinates for each of the atoms from the internal representational language model using an equivariant transformer. Okay. Okay, so they have kind of like a validation set of a couple different things here. Cameo proteins and 51 Cas14 proteins, and these are basically a holdout set. Right, the proteins used for testing are held out from those used for fitting. They're separating their test and their train, and then it's supposed that I guess for this test set they know the actual atomic structure, and then they can see how close, uh, compare the atomic level information. All right, let's look at figure 1. All right, so you have this protein here. Then you have true positive, false positive, other contacts. So blues are true positives, red are false positives. I think this is an attention matrix. So you have all the different individual parts of this. How likely are they to be in contact with the other parts? I suppose that's what it is, and that's why the diagonal is empty, because the individual parts don't contact with themselves. Predicted contact probabilities, bottom right, and actual contact precision, top left, for 3LYW. So this is the predicted contact probabilities, and this is the actual contact precision. Okay, so this is just a mirrored image. And I guess this is the actual contacts, are the grays. So I don't know, maybe they measured this in a lab. This is just kind of known ground truth. And then you can see kind of where the model is accurately predicting contacts versus no contact. A contact is a positive prediction if it is within the top L most likely contacts for a sequence of length L. Okay, what is L, right? Because if you have a classification problem with 100 classes and your top L is top 100, then it's going to always be right. So it's kind kind of a way of cheating, right? You pick an L to where your results are impressive. Performance binned by the number of MMSeqs hit when searching the training set. Larger ESM2 models perform better at all levels. The 150 million parameter is comparable to the 650 million parameter ESM1B. Okay, so smaller model, but similar performance. So this is B. So ESM1B is the first generation. MM sequence has hit with e-value less than 1. So ESM2 parameters, so this light green line is the smaller model, and then the dark blue lines are the big model. So of course the really, really big model has the best performance, but even ESM2 with 150 million, which is this line here, has about the same performance as the first generation ESM1 that had more, right? Trajectory of improvement as model skill increases for sequences with different numbers. Trajectory of improvements as model scales increases for sequences with different numbers of MMSeqs hit. MM sequences hit, so now, so this is like a these are basically, it's a 3D plot. This is the, here they're doing the number of parameters as the color. Here they're doing the MM sequences hit, and then the number of parameters is the X. So more parameters, more long range with what's P at L. Long range predictions at L top, right? Long range precision. Okay, it's precision at L. So precision is 0 to 1, right? So okay, this is a precision assuming that if the correct contact is in the top L predictions, then we're saying it's a hit, and you take all the hits, all the misses, and then that's where you get your precision from 0 to 1. Left to right shows models from 8 million to comparing the smaller model against the next larger. Points are PDB proteins colored by change in pseudo perplexity for the sequence between the smaller and larger. TM score on combined CASP14 and Cameo test sets. Points are colored by the change in pseudo complexity. Okay, so here we have in D different size models, right? The biggest one having 3 billion parameters, smallest one having 8 million parameters. Delta perplexity, the change in perplexity. Okay, so this is comparing one model to the other. Sequences with large changes in contact prediction also exhibit large changes in LLM. I don't really know what the fuck that means, to be honest. Alright, and then finally F here, we have actual stuff here. So again, perplexity lower is better, so this is the best here. Makes sense. The biggest model, 15 billion parameters, has the lowest perplexity, but we actually see the drop is quite fast. So you go from 12 to 11 to 6 to 5 to 4 to 4, right? So the 150 million parameter model and the 15 billion parameter model are honestly closer together, which to me says that the dataset size is starting to become the limiting reagent once you get to 150 million. Root mean squared difference. I don't know what RMSD means in this context, but and then PIDDT, probably some other metric. Here they're calling it PLDDT. Here it's PIDDT. 7QQA is a protein. So I think this like light gray is the actual structure of the protein, and then the colored one is the prediction. So you see that in the 8 million and the 35 million, right? It doesn't get this part here, this lower part. It doesn't predict that correctly, but as soon as you get to the 150 million parameter model, it does get it, right? And you see that it does get this, and then once you get to the 15 billion, it not only does it get it, but it gets the like kind of entrance and the exit to it quite well as well. Okay. Okay, just a bunch of different kind of ways of saying that the stuff gets better when the model gets better. Generates state-of-the-art three-dimensional structure predictions directly from the primary protein sequence. Okay, this is kind of cool. So they train ESM Fold, which I guess is their answer to AlphaFold, and this just seems to be separate from ESM2. So they're calling here a folding head. So there's a head on this model. ESM2 is really just the kind of feature encoder, and then you take that encoding and then you have a folding head, which actually predicts the folds. The sequence is processed through the feed-forward layers of the language model, and a model's internal states are passed, right? The embeddings, the representations are passed to the folding head. The head begins with a series of folding blocks. Each folding block alternates between updating a sequence representation and a pairwise representation. The output of these blocks is passed to an equivariant transformer structure module, and 3 steps of recycling are performed. What does recycling mean? And you do get confidences, that's good to know. Okay, so this is important here. This little sentence here is basically the entire speed up that they were talking about with inference, where current state-of-the-art prediction models deeply integrate the multiple sequence alignment into the neural network architecture through an attention mechanism operating across the rows and columns of the MSA. So it kind of sounds like what they're suggesting is that AlphaFold takes the entire sequence and then basically just puts all of it through a transformer. So like every part of this is paying attention to every other part of this. And they don't do that. They feed the entire thing into the encoder, and then they just feed the representation into the folding head. Right, and the folding head has a representation that represents the sequence, and then it has another vector that represents the pair. Okay, on a single NVIDIA V100, ESM Fold makes a prediction on a protein with 384 residues in 14.2 seconds. Search process for related sequences, construct the MSA. What is MSA? Did they mention that and they just like stopped talking about it? MSA alpha fold. Does MSA mean protein folding? Multiple sequence alignment, also called sequence profile, is designed to collect and align multiple homologous sequences in the key query protein of interest. Okay, I don't think these images are going to help. Maybe Google information here. Alignment of 3 or more biological sequences of similar length. Sequence structure function. An MSA is a This is a contract. Okay, that's not the right thing. This is also something different. Okay, multiple sequence alignment is really what that means. Damn, guys, we just don't really know what we're just not really good at bio here. Okay, let's look at their second figure here. Single sequence structure prediction with ESM Fold. A, ESM Fold model architecture. Okay. So we have our ESM2, which is just kind of your generic encoder, you can think of it. Feature encoder, but this is the one that's trained with the masking loss or masking task. So they take a huge database of proteins, they basically feed the entire sequence, but they mask out certain parts of it, and then they train the model to predict the missing part. So that's how this brick here is trained. But then they take that pre-trained brick, pre-trained via masked task, and then they feed it the entire sequence, and then they have, that's going to give you basically a encoded, right, some vector that represents that sequence. And then they feed that into the folding trunk, right, which is this kind of like iterative process. That's, I guess, what they call recycling. Right, so we were asking what the fuck recycling, what recycling means in this process. Alternates between updating a sequence representation and a pairwise representation. So it recycles it. Folding trunk 48 blocks, structure model 8 blocks. So predicted structure and conference. Okay, so it does a bunch of these steps here. Pair representation and sequence representation. Okay, so this is actually what the inside of a folding block looks like. So the sequence representation is what comes out of here, right? And that just represents the entire sequence. Pair representation, I suppose, is the like kind of like an attention, a 2x2 matrix where like each individual part, how likely it is to connect with each other part. I'm not exactly sure what pair. I thought initially it might have meant like two neighbors in the sequence, but I think it might be the whole thing. Okay, bias, self-attention, transition, pairwise product difference, triangular, update, transition. Okay. Okay, to the folding trunk. Okay, to the structure module, which outputs 3D coordinates and confidences. Okay, so that's what this structure module is. When MSAs are ablated for AlphaFold, performance of the model degrades. Scatterplots compare ESM Fold x-axis predictions with AlphaFold 2 y-axis colored by language model perplexity. Okay, so we have AlphaFold and then ESM Fold, so they're basically comparing their ESM Fold to Google's AlphaFold. Perplexity, again, lower is better, higher is worse. And each of these is a kind of like an individual point on the test data set. So they're showing here a lot of these points are below this line and they're blue, right? You want blue points, right? Meta wants a bunch of blue points on this side of the line, a bunch of the red points on this side of the line, which is kind of the pattern you're seeing here. Single sequence full. What is this bar chart here? Accurate atomic resolution predictions, which similar accuracy to Rosetta Fold. Okay, so this is the full atomic structure predictions, and they're just showing you that they're better than AlphaFold and Rosetta Fold. Okay, here we get a definition. PLLDT is a well-calibrated estimate of prediction accuracy. So it's accuracy. I think it's the distance between the actual, right, this like gray, which is the ground truth, and then the colored one, which is the prediction. Okay, so the little p, the lowercase p in front of LDDT is predicted LDDT, and then LDDT is the actual shape, I suppose, is a better way of, or another way of thinking of LDDT. Complex predictions on a dimer and tetramer. So these are different types of proteins. ESM fold predictions are colored and overlaid on ground truth, which is gray. Yeah, so we kind of came to that conclusion, but the gray is the ground truth, and then the colored one is the prediction. Okay, so here we have ESM fold and alpha fold. ESM fold and alpha fold, both of them kind of get the same stuff, to be honest. There is some differences, like actually if you look here, for example, we see this little tail part here. ESM fold gets the right kind of shape there. Maybe it misses this little like end right there, but for example, AlphaFold doesn't get that at all. It thinks that the interaction between these two is actually different. It's like a closed loop versus an open loop. Okay. Cool. We train the folding head on around 25k clusters, covering a total of 325k experimentally determined structures. Huh, we changed the... Let's go back to yellow. Further augmented with a dataset. That's no good, man. They used AlphaFold 2 to create a dataset of 12 million structures, and then they train on that. I would say you're introducing all kinds of errors there. Okay, so there seems to be something like multiple sequence alignment, right, which we looked up, and then there's also single sequence. Yeah, and it says that AlphaFold and RosettaFold, they don't do well with the single sequences, which is what we were seeing here, right? Single sequences versus full. So we still don't really know what that means. I think it's part of it or just like a little piece of it. Like maybe you're looking at the whole structure versus just a little piece of it. And they're saying that single sequence performance is important because whenever you do de novo protein design, which is creating new proteins from scratch, you need to be good at single sequence inputs. Pearson correlation. The relationship between perplexity and structure prediction suggests that improving the language model is key to improving single sequence structure prediction accuracy. Parts of structure with low accuracy do not differ significantly between ESM fold and AlphaFold. So that's interesting. It's saying the things that these models are not good at are the same across all these models. So it kind of suggests that Meta's models here and Google's models, such as AlphaFold, they're kind of learning the same thing, right? There's a low-hanging fruit, right? Some lower dimensional representation and understanding of these proteins that you can achieve with this rough level of scale and data set and model, and both ESM Fold and AlphaFold are getting to that same point. Heterodimeric. Heterodimeric, not comparable, is produced from two similar but different monomers. So heterodimeric means multiple dimers. Okay, so a dimer is a protein that's basically composed of two proteins. Homodimer is formed by two identical proteins. A heterodimer is formed by two different proteins. So heterodimer are basically okay, that kind of makes sense. It seems like basically some proteins, the way they work, there's kind of two pieces. There's two kind of identical pieces that fit together, and they really only work kind of in that dime, right, in that protein dime piece. But then you can also have what are called heterodimeric complexes, where it's two different proteins that also kind of tend to glue in a specific way and then be used together. RMSD 95. Okay, here we go. This is the actual definition of what their RSMD is. So root mean square deviation at 95% residue coverage. This is atomic units. I think that's what A dot means here. So basically RSMD measures kind of the distance between these atomic structures. It's a measure of how well the 3D structure is mapping to the true 3D structure, the prediction to the ground truth. I wonder how accurate these ground truths are for these proteins. Like how much mislabeled training data is there in this? And this is actually probably even a greater issue as well, because kind of what we were talking about at the very beginning here, right? It's like when it comes to like language, writing a text or using something like a GPT-3, right? Like a large language model in the space of human language, it's very easy for us to say or to evaluate that and or evaluate that model because we understand language and text so well. But um, protein folding just seems like so much less intuitive, and we don't really have a good understanding of it to begin with. So maybe if both AlphaFold and ESM2 are telling us that this protein has a specific structure and it's different from the ground truth, perhaps the ground truth itself is wrong, right? Like it would be very difficult for us to even know if the ground truth is wrong. Or maybe it's not, I don't know, but it seems like it's difficult because it's like we're not going to understand the AI here has an understanding of this stuff that we don't, so it's very hard to fact check it. Okay, so they're basically saying that roughly 1/3 here across the 617 million predicted structures, about 113 million structures meet the very high confidence threshold. So what they're saying is that we have over 617 million experimentally determined structures. Like, how good is that data set? Has someone cleaned that? I don't know. Mm, this is kind of cool. So it's saying that, right, not only does their model output a prediction, but it also has a confidence, right? So it can tell you, I'm quite confident in this prediction, quite not confident in this prediction. And they're saying that actually a bunch of the high confidence predictions are not in the UniRef 90 database, which means that it's found a bunch of protein clusters, right? Clusters in this high dimensional space of all proteins that didn't really exist. So it's basically saying many of these things that it's found are novel. Many of these structures are novel. Yeah, ESM Fold effectively characterizes regions of the protein landscape that are distant from existing knowledge. Okay, and here they're saying there's some sequences such as this one here and that one there, right? These are individual proteins that are kind of like lone wolves in the existing databases. It's like hard to say that they're close to anything else, but then once you use uh, their big model here, to predict a bunch of structures, you can actually find things that are close to it. So this is LLDT, the actual structure. This is predicted LLDT or LDDT. LDDT to AlphaFold 2 prediction. I don't know what that means. Is that the LDDT predicted by AlphaFold? Mean PLDDT. UMAP, UMAP is basically like T-SNE. It's a lower dimensional projection of the kind of higher dimensional embedding space for the sequence, right? So it's basically this here, the sequence representation. It's taking this sequence representation, which is probably not 5 numbers. It's probably, I don't know, 128 dimensional or something higher than that. And then UMAP projects it down to 2 dimensions here. And this is a way of showing you the different clusters in this embedding space, right? So this is a UMAP projection. What is this little cluster here, right? Like, what is this? Mapping metagenomic structural space. Density estimate across a subsample of around 4K sequences. So there's around 4,000 dots here. B, the distribution of mean PLDDT computed for each of the 617 million ESM fold predicted structures. Okay, so the predicted LDDT for the 7 million or 617 million different structures in the MGIFY database. So this are the 617 million here that are experimentally determined. So you mostly have high predicted LDDT, right? Which is good, but you do have some low ones here. So the fact that this is kind of like a bimodal distribution here, there's like a whole bunch where it's off by a lot here. So there's some kind of pattern that hasn't been learned yet. That's kind of what I would read from this. The distribution of TM score to the most similar PDB structure for each of 1 million randomly sampled high confidence. Okay. High confidence means PLDDT of above 0.7 and PTM of above 0.7. Closest PDB TM score. And then D. This sample of 1 million is visualized in 2 dimensions using the UMAP, colored according to distance from the nearest PDB structure, where regions with low similarity are colored in dark blue. Okay, so blue here is correct predictions, right? And then red, right, PLDDT is this blue color is 0.9, PLDDT higher higher is more accurate, lower is less accurate. So you can see that actually what often is wrong in these is like these little like skinny parts here, right? This is the red here, that's what gets predicted wrong, this gets predicted wrong, this gets predicted wrong. And maybe there's some insight there, right? I think when you look at proteins, right, usually these in biology things are kind of like keys, right, where they're like they have specific sites that fit with other sites, right? But and it always seems like the kind of these little stringy parts, those might be the sites. So maybe the stringy parts kind of change, right? Sometimes they're like this, sometimes they're like this. So maybe the fact that these models are having a hard time predicting those little stringy parts is indicative that those little stringy parts tend to change, or sometimes they're not the same, and maybe people don't realize that. Maybe in these experimentally determined structures, they just kind of like happen to say, okay, and we found out that it's like this, but in reality, in nature, sometimes that little stringy part is like this, right? And right now, we're telling these LLMs like that's not correct. It should be the first way, the part that we or the way that we experimentally determined that structure. But maybe the LLM is telling you, no, 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 no. Sometimes it's going to be like this. It's kind of interesting to think about. Okay. All predicted structures are available on this atlas, blah, blah, blah. Available for bulk download via programmatic API. Someone's paying that cost because that's going to be gigabytes of data. Brief review of evolutionary-scale language models. LSTMs, something I haven't heard of in a long time. Long short-term memory. It's an old type of sequence model that used to be popular. Several large-scale models are now available as open source. I mean, I think this makes sense for protein folding and prediction because the data sets that these are getting trained on are funded by government research, you know? These are government research data sets, NIH, right? Cancer, Alzheimer's research, all funded through kind of nonprofits. So I think it makes sense that you don't keep that separate. Let's move this a little bit so we can see better. Yeah, an era where it is possible to understand the structure of all proteins discovered in gene sequencing experiments. Right? Very high confidence. Predictions in the Metagenomic Atlas are expected to often be reliable as at a resolution sufficient for insights similar to experimentally determined structures. Active sites. This is maybe what we were trying to get at with the little section, little stringy parts. Cool. A little dense of a paper, to be honest. See if we can glean anything from the appendix here. Data, language models, ESM2, model architecture, FSM fold. I feel like this is the actual like stuff that matters here, the details. So Uniref 50 is used for training. So the training data set is from September 2021. Training data set and validation data set is around 0.5% of the training data set. Okay, so they removed de novo designed protein. So apparently there's some of those in the Uniref and Uniprot data set. They shuffle the training data, or they shuffle the training data when they put it into the batches. Okay, so each cluster is sampled evenly. So they're doing something. So sometimes when you're doing, for example, classification, you'll have a lot of examples in the training data set of one class and not a lot of examples of another class. And sometimes what people do in training is they'll, whenever they create the batches, they'll make sure that it's balanced, right? That there isn't too many examples of one class versus another class, and this will result in a more kind of robust, balanced model. And I think they're doing something similar here, where there's some clusters in these genomic databases where there's a bunch of proteins that are all very similar, and they want to make sure to sample each cluster evenly such that your model doesn't kind of overfit to a specific cluster. Okay, test set. Breaking it down into easy and hard. Chains with length less than 20 residues. What are chains and residues proteins? What is a residue? In biochemistry, a residue refers to a single unit that makes up a polymer, such as an amino acid in a polypeptide or chain. Okay, so residues are basically like the words. So maybe residue, you could think of residues as like the words or the tokens inside an LLM, or kind of in the LLM kind of abstraction. And then chains would maybe be like a sentence. So this is chains are sentences, and then residues are tokens. Logistic regression. Cij is a Boolean random variable which is true if amino acids are in contact. All right. Suppose our transformer is L layers and K attention heads per layer. Let AKL be the symmetrized and APC corrected attention map. APC corrected attention map. We don't know what APC corrected means in this case, but some kind of normalization or regularization on the attention map. For the kth attention head in the Lth layer of the transformer, and AKL for IJ is the value of the attention map at position I and J. So amino acids I and J, right, where an amino acid is sometimes called a residue. So this is the word, the tokens. So it's basically saying you have our chain, right, our sequence is composed of each of these little amino acid tokens, and then there's, you want to say, is amino acid I and J in contact, right? So if you have, let's go back to like a picture here. If you have, okay, this is better. Let's look at this picture. So what is the chance that this amino acid here is in contact with this amino acid here, right? I and J. So they're basically saying the probability of Cij, right, of the amino acid at I and J being in contact, right, where 1 is in contact, I suppose, and 0 is not in contact, which it allows you to make it a probability, and that's where you get the confidence from, right, because it's a 0 to 1. You're doing a 1 plus exponent of negative beta naught, which I assume is some kind of hyperparameter here. Where are they going to tell you where beta naught is? But, okay. Minus the sum over all L and the sum over all K, where you have L layers and K heads, of beta KL times A, the value of that attention map. Parameters are fit in scikit-learn, okay, using L1 regularized logistic regression with 0.15. There is a, oh, that's probably where they had beta naught there. Protein training set. For both training and test, a contact is defined as 2 amino acids with C alpha distance. Okay, so maybe something here, the way they define contact is kind of a binary. You either have 1 in contact, 0 not in contact, right? And then they have this arbitrary threshold of 8 atomic units as contact. I think it'd be probably more realistic, right, that in the real world, especially in biology, this is going to be analog. It's not going to be contact, it's going to be like distance, right? So maybe, right, having a distance, if you actually know in your ground truth training data set what the atomic unit distance is between these two amino acids, I feel like that'd be a more useful learning target than just a 1 or a 0 where you have a threshold. Perplexity is a measure of a language model's uncertainty of a sequence and is defined as the exponential of the negative log likelihood of the sequence. Okay, so you have some random mask M, which defines a set of tokens, right? Some subset of the input sequence. 15% probability of inclusion, so it's roughly like 10% of the total sequence. Okay, so tokens have an 8% probability of being replaced with a masked token, and a 10% probability of being replaced with a random token, and a 10% probability of being replaced with an unmasked token. The perplexity of that sequence x is thus defined as the exponent of the negative log likelihood of x of I in M. So the tokens that are masked given some tokens that are not masked, so these are the real tokens, and this is the union of the tokens that are masked. Yeah, so like this masking, unmasking, I think is basically just a way, randomly masking, unmasking. It's kind of a way of, right, for each individual token within the sequence, you have some kind of like confidence, if you want to think of it that way. Although in information theory, uh, it's more explicit. It's basically saying like, um, how much more information do I need to know the or how what is the just the probability for that individual token that we don't know? What does that distribution look like, right? Do we know a lot about that distribution, or do we not know a lot about that distribution, right? And if we take a sequence and we mask out a bunch of things, and all of those things that we masked out, we have no idea what they are, then you're going to have a high perplexity. If you take a sequence and you mask out a bunch of things, and then all the things that you masked out, you have a pretty good idea of what they are, right? That probability distribution is actually quite known. Then you're going to have a low perplexity, right? Where you understand a lot of that sequence. So it's kind of like a high level gloss over of what they're kind of doing here. And then this is difficult to calculate, or not difficult, but like masking out every combination of things in that sequence would be annoying. So basically they say we kind of do it randomly and we only do it in one forward pass, and then we call this pseudo perplexity. And this is an estimate. Yeah, so we have here perplexity number 2 compared to pseudo perplexity number 3. And pseudo perplexity is basically you're just summing this from over all tokens xi in the sequence of length L and divided by the length of that sequence, where L is the length of the input sequence. We use a BERT style encoder. Uses the original transformer paper uses absolute sinusoidal positional encoding. Okay, yeah, we learned replace this static sinusoidal encoder with a learned one. Okay. ESM2, they used a rotary position encoding. Slightly increases the computational cost of the model since it multiplies every query and key vector inside the self-attention with a sinusoidal embedding. Interestingly, it doesn't matter as much once the models get bigger, which is kind of cool, right? It's basically you have positional codings inside these transformers, right? Is stuff happening at the beginning of the sentence or the end of the sentence or the beginning of the sequence or the end of the sequence? And I think in sentences, right, it makes more sense because you have like the way sentences work, you have kind of like a time-based thing. Usually you're saying things at the beginning of the sentence kind of are a good way to know what's at the end of the sentence. But in a protein, right, the beginning and the end doesn't really mean as much because you're taking a 3D thing and flattening it out into that 1D sequence. So kind of it makes sense that position embeddings, you don't want to have this kind of like human chosen bias of like, hey, this is a position embedding that works for this, because those position embeddings are more designed for sentences, not proteins. So just let the model pick its own kind of embedding. Okay, as the models get bigger, the amount of communication becomes the fundamental bottleneck for the training speed. We increased our effective batch size. These are huge batch sizes, 2 million total tokens. Use atom optimization, just weight decay. They do have learning rate scheduling. First they do a warm-up, and then they linearly decay downwards. We cropped long proteins to random 1024 tokens. We used beginning of sequence and end of sequence tokens to signal the beginning and the end of a real protein. They use sharded data parallelism. Shards, model weights, and optimization parameters. Yeah, so when you have these huge models that they can't fit on a single GPU, you kind of have these like clever sharding ways to like put the model on a bunch of different GPUs. This stuff is there's trade-offs with this, right? The problem with this is that then you have a bunch of GPUs kind of have to wait on each other, right? And they have to, I think they say it here, the amount of communication becomes the fundamental bottleneck for training speed, right? So whenever you have these larger models that are sharded across a bunch of GPUs, every step that they take needs to be coordinated. So all the GPUs are just evaluating a small piece of the model, and then they all need to put all of those evaluations together to get to the next step, right? So that's one of the problems you have with these kind of huge models, is that they become limited by this kind of communication speed. MSAs are two-dimensional. Okay, they mask out about 0 to 15% of the sequence. So this is the folding block. We built a fault-tolerant distributed system with a main node which via TCP communicates sequences to many workers and receives result as a folded protein structures. Python dependencies. Cool. Okay, that's basically the paper. Maybe let's scroll through the code here a little bit, see what it kind of looks like. So, pyproject.2ml. Okay, that doesn't really tell us shit. Do they have a setup.py? Maybe this has the dependencies on here. Yep, here we go. BioPython, DeepSpeed, DM tree, PyTorch Lightning, ML collections, INOPS, SciPy, OmegaConf. Oh no. OmegaConf is kind of like GenConfig. It's just one of these like Python configuration management tools. They do have models trained, some model checkpoints. See these pre-trained models. How big are these? See if we can get the smallest one here. Number of layers 6. Parameters 8 million. So let's see. Okay, so the smallest model is about 28 megs. What about this bigger model? This is going to be huge. Okay, different models. Data sets. These are the specific splits they used. Citations. Inverse folding. Supervised contact prediction. Self-attention contact prediction. ESM. Okay, so you get the model. You put it on. You put it in evaluation mode, and then you put it on the GPU. This is a sequence, for example. These are each little amino acids. Make sure you're not calculating gradients. you take the sequence, you feed it into the model, you get the output. You save the output as a .pdb file. Then you use this biotype dependency to load the PDB file. And you print the PDB. You load the structure, and then you do struct b factor, the mean of the b factor. The script will make one prediction for each sequence. Interesting. They have this ESM model on Hugging Face. Okay, these are the different type of positional embeddings that they were talking about. So rotary is the one that they said they ended up using. And then they said absolute is no good. And then relative key query, that basically says that the positional embedding is absolute means like basically this would be 0, the beginning of a sentence would be 0 and the end of the sentence would be 1. Relative key means like where's the key. Relative key query is like the key and the query are different parts that are feeding into this attention mechanism within a transformer, and then the positional embedding of the key relative to the query, right? And then rotary, I think, is it's like circular, so there's just like a symmetry to it, so it's not going to be 0 to 1 for the beginning to the end. Maybe rotary position encoding transformers. Rotary embedding. Rotary position embedding. So you have position, enhanced transformer with rotary position embedding. Okay, so position is 1 through 6, right? This is the position in the sentence. So you have the beginning of the sentence and then you have the end of the sentence. That position M is multiplies the x1 and x2, which are the inputs, right? The actual tokens in the word enhanced. And transform x1 and x2 into x1 prime and x2 prime. Right, and you see this red is modifying the first word, the second word, the third word, and all the way to the fourth word. Okay, that doesn't really tell us much. ROPE are a type of positional embedding which encodes absolute positional information with a rotation matrix and naturally incorporates explicit relative position dependency in self-attention formulation. Notably, ROPE comes with valuable properties such as flexibility of being expand to any of sequence length, decaying intertoken dependency with increasing relative distances, and capability of equipping the linear self-attention with relative position encoding. So why might this work better? So if you look at a protein, right, there's some beginning to the sequence and then there's some end to the sequence. And if the distance between here, the beginning of the sequence, and something that's here in the middle of the sequence, right, that distance, right, it's like, I guess when you do the word rotation here, right, means that you go back around. So the very beginning of the sequence and the end of the sequence are somehow closer together than if you would use absolute position embeddings. Hmm. I mean, it seems like absolute position embeddings are much more popular, but I think that's also because they're way easier. Hmm. Token dropout. Okay, mask tokens, kind of like a classifier dropout. Different sizes for the attention heads, the number of hidden layers, hidden size. ESM tokenizer. PyTorch model. Cool. I mean, I think this is pretty much everything here. We're already starting to kind of run out of material, but this is a cool paper. I think it's good that we have several uh, tech giants kind of pursuing the application of deep learning to biological problems such as protein uh, structure prediction, protein fill in the blanks, and I think there's probably in both of these, I think they might be hitting the limit of the databases, right? So you're kind of getting to the point where, okay, you've kind of gotten every single protein database that you could and trained kind of the largest model that you could on those protein databases. I wonder if we're going to be able to see step function improvements from here without being able to 100x the dataset size or 100x the model size. But maybe they can synthetically create datasets with these models themselves to train even larger models. But it's an interesting problem, and I can't wait for bio to kind of get crazy, like the medicines that they will find and the molecules that they will create. Should be quite interesting. Cool. Thanks for listening, and